Okay, now we will discuss method of fish or fluorescence in situ hybridization. कि ये कैसे हम इसको complete करते हैं. It has different steps, different procedures. First of all, first step is labeling of probe. So probe which we use to detect particular sequence of DNA or RNA, that is labeled, and that is labeled with some fluorescent or radioactive tag. Generally, जो probe use किया जाता है, that is composed of few base pairs to about 1 million base pairs. ज़ाहिर है बहुत बड़ा जो साइज का प्रोब होता है वो भी हम यूज़ नहीं करते हैं. So सबसे पहले जो है प्रोब को लेबल किया जाता है. Then next step is sample preparation. So we prepare the sample. Cells, tissues, or formalin fixed, paraffin embedded, or frozen tissue sections are used. So we can use different types of sample, different types of sources. So fresh cells and tissues, they may be used to locate certain sequence of nucleotides. Paraffin embedded sections can be used, and frozen sections, frozen tissues can also be used. And these samples are permeabilized to allow accessibility of the probes. When we say samples are permeabilized, it means the cells are treated with some reagents so that the pores are formed in the cell membrane so that the probe can enter into the cell. If pores are not formed, the probe will not be able to enter into the cells, and in this way the DNA sequence will not be detected. So samples are permeabilized so that probes enter into the cells. Then the probe and the sequence of DNA, the target DNA, they are degraded. When we say they are denatured rather. So they are heated or they are treated with some alkalis so that two strands of DNA present in the chromosome in the sample or they are present in the probe, they separate from each other. We have already discussed that two strands of DNA, they are attached with each other through hydrogen bonds. Adenine is attached with thymine by 2 hydrogen bonds, and guanine is attached with cytosine with 3 hydrogen bonds. So these DNA sample is heated so that two strands are separated. Similarly, probe is heated in the same way so that two strands of probe they are separated, and when they are separated, then they are allowed to hybridize. These different steps are shown here in the diagram. First is DNA unmasking, when proteins and other molecules present along with DNA, they are separated so that DNA is unmasked. DNA is able to be detected. Then probe and DNA denaturation, it means they are heated or they are treated with some alkalis so that double-strand DNA molecule, they are separated into single strand. And then there is hybridization of probe with the target DNA so that probe, which is labeled with some fluorescent or radioactive material, that probe can bind with the DNA molecule. And then this is detected. How it is detected? If we use a fluorescent label, then it is detected with the help of fluorescent microscope, or if it is radioactively labeled, then it is detected accordingly with another detection system. And this signal is detected by using a particular type of instrument like here microscope or some other type of instrument, and this image is analyzed by different ways. So as we have discussed, the probe and the DNA sample is hybridized so that labeled probe can bind with the DNA sample. Then the probe and the DNA sample, they are washed. Why this washing step is here? Samples, they are washed to remove all unhybridized or partially hybridized probe from the DNA sample so that we don't get false positive results. ज़ाहिर अगर washing नहीं होगी तो even जो unbound जो probe होता है, जो DNA के साथ bind नहीं होता, या जो partially bound होता है, उसकी वजह से भी signal आ रहा होता है. So there should be proper washing so that we don't see false positive results. And then there is detection system, as we have discussed right now a few minutes ago, that we detect the labeled probe with the help of fluorescent microscope or some other type of detection system. So hybrids between probe and chromosomal targets are visualized by using a suitable fluorescent microscope or some other type of detection system. Sensitivity that depends on the fluorophore which we use to label the probe and the detection system like the fluorescent microscope. If we use simple fluorescent microscope, there will be limited sensitivity. If we use confocal microscope, for example, then we will have better signal from these lights.